What I'm talking about today, much focusing on the molecular genetics of recent PEDV isolates and understanding the variability of PED virus. This is a typical clinical sign of PED outbreak and the gross lesion. The severe vomiting and dehydration as watery diarrhea and the feces-coated skin is observed, will be observed during the PED outbreak. The transference of small intestine and yellow granule deposition in kidney cells. In most cases, this kind of flatbedding or harrowing bond is a facilitate of PED virus and some enteric pathogens. In most cases, endemic swine farm holding PED virus in the growth finishing premise, just like this kind of facility holding PED virus in the contaminated feces. We can completely clean with this kind of facility to remove PED virus. In Korea, we don't have any PED or TG outbreak this year. Due to the year 2010, we have a severe FMD outbreak. We are stamping out about 40% swine population of a total number of pigs. That time, we have a very routine disinfection policy every Wednesday, so we don't have any problem now. But 2007, we have about 36 PED outbreak. That time, we have really suffering from the PMWS, circovirus-associated disease, and the PRRS. So I think PRRS or porcine circovirus infection affect PED vaccine efficacy. In most Asian countries and Korea, we have 4 major swine disease: classical swine fever, porcine circovirus type 2, PRRS, and PED. In most cases, we have a very nice vaccine program, classical swine fever. The modified live vaccine very effective to control this disease up to 95%. After development of porcine circovirus vaccine, we can control very easily with mass vaccination through the government support. Still, we have a PRRS problem, but after FMD outbreak, we have different genotype, especially European type is prevalent at this moment. European genotype is much less severe compared to the North American PRRS virus. Routine seroprofiling of serum-neutralizing titer in the commercial swine farm suffering from the endemic situation. We have 2 nice peaks in the breeding population, including sow and 130 days old growth to finish area. The PED viruses belong to Coronaviridae family. Extremely difficult to differentiate from TGE by clinical sign, but serologically unrelated with TG virus. We have very severe problem early of 2000. Our laboratory start January 2002 through 2007, almost 5 years duration. We collect 1,932 samples from swine farm. Just we have 351 positive results by RT-PCR assay. That time, we like to know about some sequence information through the S gene, ORF3, and M gene. And also, we have a trial to isolate virus with field samples. Just selective way, we put in 35 isolates. Finally, we can successful isolation with 10 isolates. During this study, we have very difficult technique to isolate the PD virus within less than 5 passages. We lose about 2/3 samples through the tissue culture passage. But after that, the virus isolation was actually stabilized through passage level 10. Through the spike gene analysis, we have 3 different groups identified genetic base G1, G2, G3. The G1 was subdivided into G11, G12, and G13. In most attenuated vaccine strain belong to the G12. Another ORF3 analysis. ORF3 play a role for attenuation or virulence factor. So we have the same pattern of spike gene. We can classify the 5 different branches. M gene also analyzed by sequence analysis. We have 2 major branches, including the upper one has 3 sub-branches, lower one has 2 sub-branches. Very recently, we have field isolates analysis with Chinese strain or Korean strain. We classify again group 1, group 2, and group 3. Group 2 belong to the most attenuated vaccine strain based on the full-length E gene. The homology between the amino acid or nucleotide, very high homologous pattern, over 95% in nucleotide base or amino acid base. In this study, we can conclude recent PD isolates more close similarity between China, Korea, and other countries. Last year, Chinese people published one interesting paper about PD outbreak. That time, they are supporting the Chinese strain is highly close to the Thailand in most Southeastern Asia. The pathogenicity was very similar in most virulent virus. They lost over 1 million piglets in 10 provinces. The clinical sign reached to 100%, lower 60%. The mortality will be 50 to 100% range. In this report, notified they used divalent inactivated TG or TG and PD vaccine. At this point, depending on my experience, inactivated PD vaccine does not work very well all the time. So we have inactivated vaccine in Korea developed by the government institute. Still, we have questioned the efficacy in the field situation. I will show one slide at the end of this lecture. This study showing one notable finding with milk. They detected PD virus through the milk by RT-PCR assay. This is actually post-report finding of PD detection in milk. They supporting the Chinese strain is highly close. The Thailand strain, 98.5% through 98.9% similarity. But the old reference strain is a little less homology, 88.7 to 98.9%. In China, they developed most inactivated PD vaccine with prototype strain CB777. This is old strain. But the similarity will be dropped into 93.3% through 94.7%. Last year, Dr. Song and me, we requested by journal office to publish review paper. We cover molecular epidemiology, diagnosis, and the vaccine of PD virus. As you know, most coronavirus has 3 non-structural protein and 4 structural protein. Non-structural protein ORF1A, 1B belong to the polymerase activity. ORF3 I mentioned already, the virus attenuation or virulence factor. The major spike protein function as neutralizing activity and receptor-mediated virus entry into the cell. This review covered whole phylogenetic analysis in most East Asian isolates. The prototype CB777 is a black color. It's go together with most Korean, Japan, and early Chinese isolates. In Korean Peninsula, we have one island, the mainland, typically develop a kind of PD virus evolution. This evolution is highly involved with Chinese outbreak. Recently, Chinese outbreak high close relationship, the Vietnam strain or Thailand strain. The Thailand strain is green color, Vietnam strain is violet color. The Chinese is red color. The most Korean people like blue color, so we use the blue color in Korean isolates. So actually many publications support our review paper. The most Asian recent isolates originate from Korean Peninsula anyway. But one research team in China, they published a kind of idea, new variants from PD virus identified in vaccinated swine herd through the less efficacy of vaccination. The clinical sign and the gross lesion histopathological finding was very similar in previous reports. They have very severe vomiting and dehydration and the transparency in small intestine and villus atrophy and vacuolization in villus. They made some phylogenetic tree. Their new variant isolates made another cluster in center with black dots. But the upper area, we have attenuated DR13 isolates with black dot 2 new isolates. They supposed maybe these new variants came from the combination of wild-type virus and vaccine virus. Phylogenetic analysis of complete M-gene showed all South China PED strain have close relationship with Korean strain and Thailand. I supposed Thailand and Vietnam strain highly correlated with the Chinese strain, but differ genetically from the vaccine strain CB777 prototype in European strain. Another phylogenetic analysis based on the S1 and the SM gene, which have high level of variation, indicating each sample were related to a specific reference strain, and this finding was consistent with protein characterization prediction analysis. This is actually a new finding with correlation between the nucleotide and protein prediction. Also, they have group 1 and 2, group 1 subdivided in subgroup 1 and 1, 2, 3, and another subgroup, group 2, subdivided into subgroup 5. At this moment, I like to think about the PED variability compared to the immune variability in individual pig. The PEDB immune status in endemic pig farm is highly different with virus variability. First time we tried to find out the serum neutralizing titer distribution in different parity sow. They showed like this: parity number 1, 2 until 5. Few sows have very high serum neutralizing titer. Even others have moderate or low titer. So another time, during the 2007 PD outbreak, one swine farm, case number 999, we designate very random way, SN titer distribution was very typical to the virus existence in the premise. Circling period, we have very variable susceptibility with the virus, but after using feedback method, the most virus exists in the growth to finishing area. They still stimulate some pig to produce serum neutralizing antibody until marketing age. So actually we tried to identify the marketing age SN distribution with 20 animals. Some animals still exposed to the wild type virulent virus. They show 512 SN titer. The other pig population showing moderate or lower zero profiling. This slide gave me a very good idea for development oral vaccine of PED. The PED virus can impact enterocyte in the villus. They proliferate very well and then stimulate M cell in the enteric immune mechanism. Antigen presenting cell induce some antibody production through the mucus-associated lymphoid tissue mechanism. Secretory IgA released from the villus, and then this secreted IgA covered with villus to protect further PD virus infection. Another experiment, how distribute PD virus, different immunization method. So we have a different pattern in intramuscular administration compared to the oral administration. Most tissue distribution was very similar without lung. Only intramuscular administration has distribution of lung PD virus existence by RT-PCR assay. Another point was oral immunization method can induce very quickly 2 days after inoculation through 6 days. But intramuscular injection a little late, 4 days after injection, post-injection through 13 days DPI. And also we analyzed some cell affinity through the FACS cell analysis. In the intramuscular administration, they can stimulate B cell population until 20 days post-inoculation compared to the control animal. So, oral administration, how to different from intramuscular administration. As mentioned before, the intramuscular administration can stimulate the B cell population for the 20 days post-infection, but the oral administration can stimulate CD8 population until 20 days post-inoculation. So, through this experiment, intramuscular inoculation most focusing on the humoral immunity, but the oral administration will triggering the CMI much more than intramuscular administration. This is a quick tool for detection of the PD virus. In Korea, most swine veterinarian and farmers using this kind of kit. This kit costed $10 per test, but only 10 minutes you can detect the PD virus existence in the feces. After finding severe vomiting, they try to detect PD virus with this kit and then call me to for the assay feces by RT-PCR. We can support within 24 hours after receiving the feces through the RT-PCR assay, we can support the prevention pharmacy too in the swine farm. We have many different ideas of the role of PD virus shedding with oral inoculation of DR13 virus. The different age group show different shedding pattern. 3 days old piglet shedding virus up to 6 days, 2 weeks old pig 9 days, most adult pig shedding only 3 days. And also we like to observe the immune stimulation with the special immunoglobulin against the different administration routes. In most cases, oral administration really greater induce of IgA antibody compared to the intramuscular administration. Many different farms showing same pattern. Just I'll show the two typical farm finding. And oral administration is not difference between the induction of IgG or IgA immunoglobulin level, but the intramuscular administration has distinct difference. IgA induction was a little lower than the IgG induction. This is the same thing after colostrum intake. During the outbreak, we are thinking about how to circulate the PD virus in the littermate. So we tried 3 generations of virus shedding patterns in the serial passage of colostrum-deprived pig. We have further passage getting shorter shedding period. The first time we have 6 days shedding pattern, but the second passage we have 4 days only. Another third passage we have 3 days only. We didn't continue to find when they stop spreading. But this kind of study, it takes about over 2 weeks. So in most cases, PD virus can make a greater loss less than 10 days old piglets. So we didn't need to continuation of this kind of study further the long time. So in most final slides, this is a survival rate of challenge study. The oral administration could survive 85% challenge piglets compared to the IgM administration only survive 40% vaccinated piglets. This study just the vaccine applied to the pregnant sow, and then after colostrum intake, we challenge the wild-type virus. So this is a practical way we are using PD vaccine, the late pregnant sow, in 2 injections. In most oral administration is more effect compared to the intramuscular administration. I have 3 PhD students, Dr. Oh, Song, and Park. They have different function. Dr. Oh developed IgA ELISA kit, and Dr. Song developed oral PD vaccine development. Dr. Park recently graduated from under supervision of my laboratory. He studied much focusing on the phylogenetic study on molecular epidemiology. During my PhD study, Green Cross Veterinary product funding almost 10 years until now they going to expand their business in the Eastern Asian country. Still, I have a question about many research groups argue and claiming the cross-protection of different genotypes. In the previous study, we have almost the same result between the different genotypes, but we have a problem. We produced antibody against each genotype, but the antibody titer was not good. In most cases, the commercial piglets can induce less than 10 SN titer with our immunization. So the comparison was almost the same between the fourfold, less than fourfold difference. So we are trying again with different way to observe something different, the genotype cross protection. So at this moment, we are thinking about new vaccine candidate or cross-utilization study with different research systems.